And our next speaker is Natalia Freund from the Faculty of Medicine here. The floor is yours.
Okay, thank you. Can you all hear me? Cool, so thanks a lot. I'm very excited, and thank you for the organizers for this opportunity to present our work. And as can be understood from the title of the presentation, and also for those of you who know me, I will talk mainly about antibodies. And antibodies are produced by B lymphocytes, which are components of the adaptive immune system. And as such, they are able to specifically respond to infections and vaccinations. And how do they do so? So B cells present in our body in the form of naive B cell repertoire, which is an enormous collection of different B cells. What does it mean, different? So the B cells present different antibodies on their surfaces. And once this naive repertoire encounters an antigen, whether it comes from an infection or from a vaccine, there is an activation of several B cells that are able to specifically recognize this antigen through their membrane antibodies. And once this occurs, the B cells become activated, they start dividing, and then they start migrating to the secondary lymphoid organs and form, together with T cells and dendritic cells, these unique anatomical sites that are called germinal centers. And in the germinal centers, affinity maturation takes place. B cells mutate their antibody genes to further diversify their ability to recognize the original cognate antigen. They divide, and after several rounds of division and mutations, they go to the other compartment here in red, where they meet the dendritic cells and T cells, and they take a test to be selected for the ultimate best B cell that binds the antigen with the highest affinity, but also presents other traits such as antibody stability and the ability to not recognize any autologous antigens. So only these B cells are able to proceed to the next stages. And there are two main products of cells that are exiting from the germinal center. The first product are antibody-secreting plasma cells, and these are responsible for the antibodies we see in the serum. And the second product are memory B cells. And memory B cells, they exit the germinal center as a clone. What does it mean, a clone? They all bind the same antigen. They all present antibodies against the cognate antigen. However, their antibodies are slightly different from each other, and that is in order to be able to accommodate in the future variations of the antigen that is slightly different from the original antigen that was recognized. So these memory B cells, they stay for very long periods of time in our body. And they are, in fact, the basis for what we call immunological memory. And in fact, we have many different clones of B cells in our body, of memory B cells, every clone responsible for a different antigen, for a different pathogen, and in a way that this compartment collectively represents a footprint of past and present infection status of every individual. So we are especially fascinated by this compartment, and the goal of my laboratory in Sackler is to try to decode the memory B cell repertoire in a way that we can understand what B cells are responsible for which diseases, and what is the absolute best solution of the immune system in terms of antibodies to tackle different diseases. So we call it the learning from the body approach. And the idea is actually quite simple. We recruit individuals who have a certain disease profile. We collect their cells from a blood sample, and then we try to isolate specific B cells with disease-related proteins that we call baits. We use them as a fishing pole to pull out the B cells that we are interested in. And then we sequence the single cells, we sequence the heavy chain and the light chain for every cell. We analyze the sequences that were produced by the immune system in response to an infection or a vaccine. We can select antibodies that we would like to continue working with. We can clone them to expression vectors, produce them in the lab. And in the end, we have what we believe is the natural antibodies that were made by the immune system, by the germinal centers of these individuals. So we applied this platform for several disease models. For HIV during my postdoc at the Rockefeller University, we did a work on tuberculosis that was published last year. This is Aviv Watson's project. And 2 years ago, when COVID-19 arrived to Israel, we were eager to try this platform to characterize the antibody response against the new virus. So this project was all and is still being led by Michael Moore, who is a very talented PhD student in the lab. And the dog's name is Johnny. And Michael did his master's in Johnny's lab, not the dog. So, yeah, thank you. So the first question that we asked was what antibodies are developed in response to infection, when we were particularly interested in whether there is a difference in the antibodies that are produced during severe COVID infection and asymptomatic COVID infection. And we thought at the beginning, well, obviously antibodies are so important, so people who have asymptomatic infection must have some pre-existing antibodies or something that protects them. So we focused on the RBD, the receptor binding domain, which is this component of the spike protein. The spike protein is the protein that is presented on the outside of the virus, and this is the component that binds ACE2. And ACE2 is the receptor of SARS coronavirus, which is the first entry point into the cell. And if we look on the protein-protein interactions, this is how it looks like. The RBD is in orange here. ACE2 is in green, and this structure was solved shortly after the virus was identified. And we collected a cohort of 18 patients. It was the very first cases of COVID in Israel. Some of them were severe infected, others were mild infected. They were all the same age, all healthy, and all 6 weeks post-infection. And we did ELISA with RBD. And what we found was first that we were really deeply wrong. Those people who had mild infection had significantly less antibody responses against the RBD. And not only that, they could also inhibit less the interaction between the RBD and the ACE2, this interaction, as opposed to severely infected, all of which were hospitalized for at least 1 week in the ICU. We then sequenced the B cells of our donors, of 13 donors. We did it by a single-cell BCR-seq using the 10x genomics platform. This was done in collaboration with Immune AI company. And we could see that severe donors had also greater stimulation of their B cell compartment. They had more clones. And also they had one VH gene that stood out, and this was the VH gene 3-53 that was also at the same time discovered as related to neutralizing response against coronavirus. So severe donors had probably more viral stimulation as a result of their disease and the inflammation, and therefore they had a better B cell response. We also did single-cell sorting using RBD as a fishing pole to fish out the memory B cells, and we cloned many antibodies, or Michael cloned many antibodies, and 9 of the antibodies that he cloned turned out to be neutralizing. Now, how do we know that they're neutralizing? We did 2 different assays. The first assay was a pseudoviral assay that was done in Tzfat by the lab of Meital Gal-Tanami and Moshe Dasau. And the second assay was the live viral assay, and this is the only one that I will show. Can we? Yeah. Thanks. So this assay was done in Vero cells, and you can see here the controls. It was done in San Diego by a collaborator, Ben Crocker. And when the cells are healthy, there is no virus, you can see the blue staining. And when we infect them with the live virus, we can detect this red staining. And 2 of the antibodies that we tested were really weak neutralizers. So you can see a significant difference from the control in this concentration. We needed very high concentrations of the antibody to see an effect. However, luckily for us, the other 7 antibodies were pretty good. So 4, we call them moderate neutralizers. And this is because we could see still some virus left. At this concentration, this is a 10 microgram per ml antibody in this culture. However, clearly the antibodies were doing something. The other 3 antibodies, we call them exceptional neutralizers because in this concentration and even in lower concentrations, they could completely clear the culture. So we did a couple of ELISAs, and we could see that all the neutralizing antibodies could bind RBD and spike in ELISA. This makes sense, with the exception of one antibody. It's here, this orange one here, and we will speak about it later because this antibody is also one of the exceptional, really good neutralizing antibodies. 4 antibodies were able to inhibit ACE2 binding in ELISA, and when we repeated this result with flow cytometry, we expressed the whole trimer on cells, and we tested whether the antibodies could inhibit ACE2 binding to the spike. We could see that the same 4 antibodies were very effective in blocking ACE2 binding to the cells, and also this tau2212 antibody that did not bind in ELISA was actually very good in inhibiting ACE2 binding to spike. Other 2 antibodies, 1109 and 2310, were neutralizing but via a different mechanism because they could not block ACE2 binding to spike. So we will get back to that. So the paper was published last year, and around this time vaccines became available, and the whole world entered a global vaccination campaign, as we've heard a lot this morning. However, as we understood, the virus was fighting back by production of variants. Here you can see the variants across the timeline in the US. This is from our world in data. So we were very curious to find whether our antibodies that were isolated from individuals infected with the wild-type virus can also get the variants. So you can see that wild-type and alpha are very similar, and this is binding data for RBD. However, when we test the beta, gamma, delta, and omicron, we can see a gradual loss in activity. And this is especially obvious for omicron, where the antibodies from the class of ACE2 binding site are no longer effective. And these are the most potent antibodies that we have. The antibodies from the non-ACE2 binding site group are still binding, though. But what about neutralization? So we did the pseudovirus and the live virus assay. I will show only the live virus assay for the lack of time. So these are neutralization graphs. The y-axis presents area of infection. So the lower the bars, the better the antibody. And you can see that most of the antibodies neutralize the wild-type strain. These are our top 3 antibodies. They're getting the wild-type really good, reducing infection almost by 100%. And we can see that when we look at beta, gamma, and delta variants, we start to see some loss of activity. And when we look at omicron, no activity at all. So we were wondering, what is the mechanism? And of course, it has a lot of implications to people who were infected with wild-type virus. And now, how can they be infected with omicron? Well, they can. Now, we focused on one antibody. This is antibody tau2303. This was one of our best neutralizing antibodies, and it also was the only one in its group that could neutralize the Delta variant very effectively. So it was interesting for us. And how do you study protein-protein interactions? So the best way to do so is to crystallize the antibody with the antigen and look at the atomic level on the interactions between the 2 molecules. So luckily, we had an ongoing collaboration with a group in Tsinghua University in Beijing on our tuberculosis project. And they were very eager to try our antibodies and crystallize them with the RBD. And luckily, they managed to crystallize the antibody with the RBD. And you can see it here. This is X-ray crystallography. The resolution is 2.42 angstrom. And you can see here 2 structures, one superimposed on top of the other. The first structure is the tau2303 antibody with the RBD in orange. On top of that is the structure I showed you a few slides earlier of the RBD in orange and ACE2 in green. And what you can appreciate is that both molecules are binding almost to the exactly same surface. And this is the mechanism for antibody neutralization, of course. Now, if we look at the interface here in yellow, we can see that the mutation of the alpha strain is in the interface. However, the affinity is so high, the antibody binds at a nanomolar range affinity, that it doesn't affect very much the activity. Same is with the gamma and delta. Same with the beta and gamma, and the delta strain has several mutations, but luckily none of them are in the interface of the antibody. Therefore, there is no effect on antibody activity, and the neutralization is similar to the wild type. However, Omicron changes everything, 7 mutations, all in the interface, and the antibody loses its ability to bind. Now here I just want to know that the virus here very elegantly manages to mutate only the residues that knock out antibody binding, because the affinity of the RBD of Omicron to ACE2 is actually higher than the affinity of the wild type to ACE2. So this is, I think, pretty neat. Now, I want to focus on the other antibody, and this one is really interesting because this antibody didn't bind any of the antigens in ELISA. I showed you a few slides ago. And the structure here again, we were lucky to get a cryo-electron microscopy structure of this antibody with the spike. And the structure really gave some answers about why, and you can see it here. This is the spike trimer in 10, the RBDs are in blue shades, and the antibody is in dark pink. And this is a 3.8 angstrom resolution. This is high-resolution cryo-electron microscopy image. And what you can appreciate if you look on this structure from the top, that the antibody is actually recognizing a quaternary conformational surface that includes 2 RBDs in a head-to-tail conformation. And this is why it doesn't bind any one single RBD in ELISA. Now, this antibody, as opposed to the previous one, does not bind to the exactly same surface as ACE2. However, the fact that 3 Fabs are able to bind the RBD from the top, they block the accessibility of ACE2 to the RBD, and therefore they block receptor binding. Now, again, if we investigate the interface, as you can see in yellow here, the epitope, the alpha mutation, the alpha strain mutation is outside the epitope. However, the beta strain and gamma strain have the 484 mutation, which by itself, single point mutation, is sufficient to knock out antibody binding. This mutation is absent from the delta strain, therefore the activity against delta is the same, it's still active. However, again, Omicron, 35 mutations in spike, and of these mutations, 4 are in the interface, and therefore it completely escapes the antibody. Okay, but these are ACE2 binding site antibodies. We have other class of antibodies, luckily, that are less potent. But these antibodies neutralize all variants of concern. These are antibody tau 1109 and tau 2310. So how do they do it? Okay, so as usually in science, the most interesting antibodies are the ones that you can get structure of. And our collaborators were unable to get any crystals of these antibodies. So we turn out to what we know, and this is epitope mapping using phage display random libraries. And this was done by Rimo Watad, who is a rotation student working with Michael, and together they screened the antibody with random phage displayed peptide library provided by the laboratory of Johnny. And they were able to isolate many peptides. And these peptides didn't match to anything linear in the RBD sequence, but they all had very high concentration of aromatic residues and hydrophobic residues, and also asparagines. So here, Ksenia Polonsky, who is a master's student in the lab, joined a Safra computational student between me and Professor Tal Pupko. She used the Mapitope computational algorithm prediction that was also developed in Johnny's lab several years ago. And she was able to focus on this epitope here. You can see it in purple, and you can really see that, first of all, it's distant from the ACE2 binding site, and it contains these beautifully stacked aromatic residues. So we weren't convinced, so we tried another method. So Ksenia's project deals with AlphaFold algorithm that was developed by Google and published. And what she did, she used AlphaFold to predict the structure of 1109 antibody and then computationally dock it on top of the 3-dimensional structure of the RBD. And she got the prediction. You can see here the predicted structure of the antibody bound to the RBD. And in blue is the Mapitope prediction. And surprisingly, both predictions matched exactly to the same place. So now we're optimistic, and we have to confirm it by point mutagenesis. So I'll summarize quickly. I showed you that severe COVID is associated with better B-cell responses compared to mild COVID. I showed you that in a way, in COVID, it looks like potency and breadth are one at the expense of the other. So the antibodies that are more potent might be more strain-specific, unfortunately. I showed you two crystal structures of two antibodies that block ACE2 binding by two different mechanisms. And we discussed a different class of antibodies that bind in a less immunogenic site on the RBD, because these antibodies are clearly a minority. And however, they are more broad and might be a better fit for future therapies. So I'd like to thank the people who did the job. All this project is led by Michael, who is here, and also with the help of Rima, his student, and Ksenia, who did the bioinformatics, and all the other team members of my amazing lab. I couldn't be more proud. Also, we had help from the Gershoni lab, from Bar Ilan medical faculty in Sfat, and clinicians who helped recruit the patients, and of course our collaborators across the sea from UC San Diego and from Tsinghua. And I will be happy to take questions.
So unfortunately, we're running out of time, and so you'll have to grab Natalia in the break to ask questions. And